Thank you all so much for joining us for our April Town Hall. We're so grateful to see so many of you joining us from all over the world. I think this is probably the most time zones we've ever had represented on a town hall. So whatever time it is for you, thank you so much for taking time out of your busy day to join us. It's great to see so many members of the community coming together. We've got families on the line. Hi, Annie. Hi, Brenda. Hi, Steve. I see you all. And we've got some amazing supporters as well. Jeff, I see you. Thank you for joining us again. And some folks from the industry and the medical communities as well. So welcome, everyone. For this month's town hall, we're here to focus on EB Simplex and a really exciting clinical trial that's taking place with sites all over the world to evaluate a potential simplex treatment. We're honored to be hosting this town hall in partnership with TWi Biotechnology, and we're so grateful to their whole entire team for everything that they do to support and make a difference for this community. Today, we're honored to be joined by representation from TWi, a key opinion leader working on their phase 2/3 clinical trial, as well as a dedicated EB and rare disease patient ambassador. You'll hear from Nisha Trivedi, not only about her personal journey with EB Simplex, but also about how she's using her voice to advocate for the rare disease community at large. You'll hear from Dr. Yiumo Michael Chan about TWi and their EBShield study. And lastly, from Dr. Johan Bauer on his perspective on the trial and the science behind it. So just to provide an overview of the format today, we ask that everybody please stay on mute to respect our amazing speakers. And just as a heads up, we're going to save our questions for our Q&A session at the end after all of our speakers have presented. And while we'll prioritize those questions that were pre-submitted when you registered, we'll do our best, as always, to answer any outstanding questions, whether they come in live or through the chat. And if your question is not answered, we will follow up with you personally afterwards via email. So feel free to send any questions as they arrive in the chat, or if you prefer to send them privately, you can message Margot Sosa via Zoom on our team. Before I pass it over to our incredible speakers, I wanted to just start with a little bit of context on why we're here today and give some background on EBRP for any new faces in the crowd. EBRP is the largest global nonprofit dedicated to funding research to accelerate treatments and find a cure for EB as quickly as possible. And over the past few years, we've made some critical shifts in our research priorities to help us achieve that bold goal. So when the organization began, we focused a lot of our funding on RDEB research specifically, just sort of by the nature of how we were started. But thanks to initiatives that were started and funded by some of our biggest supporters, shout out to the Desha family, we've expanded to fund some of the best science across all and every subtype of EB, including a focus on EB Simplex in recent years. We funded a lot of really innovative and exciting work in Simplex, really everything from protein therapies to organoids, and we're so grateful to companies like TWi that take this science and start to bring it into the clinic, because that's when it gets into the hands of patients who need it. Another shift within how EBRP funds research is that we now prioritize research applications that will have a significant impact on the quality of life of those living with EB, or are by medical definition curative projects. And we do aim to prioritize work that will be commercialized or in the hands of patients in the next 1 to 4 years. Excitingly, we actually have a research cycle that's under review by our scientific advisory board right now. And for the first time ever, over 40% of those applications that we received in that cycle were specifically to advance curative work, which is something we're really excited about. Approximately 90 cents of every single dollar that's donated to EBRP goes directly towards funding this kind of research, and we've raised over $70 million to invest in more than 160 innovative EB research projects. This work and what we have told the research community are our priorities has directly led to a transformation of the EB landscape, from 2 clinical trials when we started to now 50, over half of which EBRP has directly funded. And this is actually the first time we get to say this, from 0 approved treatments to 3 FDA approved treatments. Just quickly, for anyone who might have missed the big news yesterday, Abeona Therapeutics' Zevaskin, an autologous cell-based gene therapy for the treatment of RDEB, was approved by the FDA, and EBRP is so excited about this because of what it means for our community, but also what it means for our sustainable venture philanthropy model, because we're very proud to have been an initial investor in this work. And we're so excited for the continued momentum and the new hope that this brings. But like I said, all of this, momentum, it's really exciting, and it would not be possible without so many of you on this call. But But that momentum and these milestones, they're just that. They're stepping stones on our journey to a cure. And our team is more laser focused than ever on advancing more treatments, especially for additional subtypes like junctional EB and EB simplex, and of course, ultimately on delivering a cure. It is now my pleasure to introduce you to our amazing speakers today. First off, we're honored to be joined by Nisha Trivedi, a selfless and tireless leader in the EB community and a passionate advocate for rare disease at large. Drawing from her personal journey with EB Simplex, Nisha's advocacy work includes sharing her story with members of Congress to promote policies benefiting rare disease patients, serving on EveryLife's Foundation's Community Congress, and on the Rare Disease Legislative Advocates Advisory Committee. Also with us today, we have the wonderful Dr. Yiumo Michael Chan, a senior consultant at TWi Biotechnology. Dr. Chan actually discovered the genetic basis of the EB simplex Weber Cockayne subtype during his graduate study at the University of Chicago, and he has since demonstrated a successful track record in rare disease research and has led multiple preclinical and clinical programs in the US and in Taiwan. Prior to his transition to industry, Dr. Chan held a faculty position at Geisinger Medical Center, and he is also serving as a scientific advisor in several patient advocacy groups. Last but certainly not least, we have the amazingly talented and dedicated Dr. Johann Bauer. Dr. Bauer is a professor and the chair of the Department of Dermatology at the University Hospital Salzburg and director of the EB House in Austria, which is the National Reference Center for EB in Austria, although they see patients from all over Europe and frankly the world. He has a long-standing career in molecular diagnosis, clinical management, and research on skin biology, blistering diseases, skin aging, and neurobiology of the skin. Dr. Bauer's primary interest is to develop novel therapies for patients with EB, with a particular focus on anti-inflammatory and gene therapy strategies. He has led numerous international clinical and translational programs and played a pivotal role in advancing investigational therapies from the bench to the bedside. He has authored over 150 peer-reviewed publications and served on the editorial board of the British Journal of Dermatology, in addition to advisory roles with DEBRA International, the European Academy of Dermatology and Venereology, and other organizations. So a huge, huge, huge thank you to all of our speakers for everything that they do for our community, and of course, for taking the time to join us today. With that, it is my pleasure to pass it on over to Nisha.
Great. Thank you so much, Allison. Really appreciate it. And yeah, today I'm really excited to share with you my journey of living with EB Simplex and my journey with advocacy. Great. So to tell you a little bit more about me, again, my name is Nisha Trivedi. I live in South San Francisco, California, so in the Bay Area, pretty close to SFO Airport. In terms of my professional life, I work as an MBA admissions consultant. So that means I help candidates to top business schools in the US and abroad with their application strategies. I also have a small side business as a business communications and etiquette trainer. So that's what I do for work. And then in terms of my personal interests, one thing I really enjoy living in the Bay Area is wine tasting. And Allison and I actually participated in an EVRP fundraiser with the wine tasting theme back at the end of 2023, which was really fun. I really enjoy travel. EB makes that challenging, but I definitely try my best. I picked up cross-country skiing a few years ago. Still mastering the art of that, and that's another thing that EB makes challenging, but I found ways to make it work. And I also really enjoy being a patient advocate for EB, and I'm excited to talk more about that today. And so in terms of my EB diagnosis and my journey, I was diagnosed with EB simplex at birth. So shortly after birth, it was seen that, there was a little bit of skin missing, and I was immediately taken to the, bigger hospital in the region, where it was confirmed that I did have EB simplex, but of course not the subtype. I was born, in the 80s, and there wasn't a lot of information about genetic testing and that sort of thing back then. Much later, in my mid-20s, I did have a genetic test, and that is when my specific genetic mutation for EB simplex was confirmed. And it was known that I had what's actually a recessive form of EB simplex. It is a mutation of the KRT14 gene, and that is actually rather unusual. As you might have heard, EB simplex is generally autosomal dominant. But in my case, each of my parents are not affected by EB. They each carry the recessive gene, and they had a 25% chance of passing it on. And I was born with this form of EB simplex. And I would say that it probably falls in the generalized intermediate to severe end of the spectrum for EB simplex. And in terms of the challenges that I've had of living with EB simplex, I will say that, with EB simplex, of course, it does tend to be on the milder form of EB, compared to other subtypes such as RDEB. But it definitely has its challenges. My skin is extremely fragile. I get blisters and wounds from very minor friction. For example, I've been on a conference call where I've, lightly bumped my hand against a table and I've gotten a blood blister. I'll, get a wound from just, bumping into something. The tops of my feet tend to blister very easily, so I have to be incredibly careful about shoes. And I just have to be, I feel at all times hyper vigilant. That makes sense. So I don't have a ton of wounds on a regular basis, but I get blisters on a daily basis. And I know that the potential for wounds is always there if I were to, let's say, have an accident, trip and fall down. So I always have to be extra, extra careful about any sort of daily activity, including walking around my own house. One of the challenges, and actually before I get to the second bullet, I'll also say that in addition to the skin challenges, there's also oral involvement with my form of EB. I get blisters on my mouth and my tongue on a semi-regular basis, and that can, of course, be challenging as well with eating and speaking. And then the second bullet point is that, of course, growing up, I had this difference from family members and peers. Again, since my form of EB is recessive, I am the only family members who's affected. And I grew up in a small town of about 10,000 in rural Pennsylvania. There were already differences, right? I was one of the few, South Asians in my town. I was, kind of pinned as a nerd pretty early on in my educational career. And then EB was just another difference. So that was difficult. And as I was growing up, and maybe even a little bit today, I received invasive questions about my appearance, and I would have to figure out how much detail, were they really entitled to. And I always had the constant need to explain my physical limitations. So from, being a child in gym class, to now having to explain to friends why a 3-mile hike is not a good idea. So those are some of the challenges that I, have definitely grown up with in the condition. And, when I was growing up, a lot of my focus had been on almost, hiding my condition. As it became a little bit less visible over the years, I would, find myself, really dodging any questions or just not bringing it up at all. And, back then it was, the 80s and 90s. It was before social media. And, the focus again had been on, telling as few people as possible. But then in my adulthood, something started to shift. And I realized that there were, definitely opportunities to bring a spotlight to this condition, to help others. And there were a few things that inspired me. One is that unlike when I was growing up, social media, obviously has become a thing. And I saw a lot of people being open about their own conditions, rare or otherwise. I saw people in the EB community, many of whom are a fraction of my age, sharing their story. And then I thought, if they can do it, maybe I can too. And so the first step I took was, this was in, I believe, fall 2019, right before EB Awareness Week, which takes place every October. I shared my story on social media. So I went from, hardly anybody knowing about my condition to, my entire network and beyond. And the response I got was very encouraging. And so that's what began my advocacy journey. I attended the Rare Disease Film Festival also in fall 2019, and I actually had a small feature in a film called Bad Days, Good Days, and Rare Days, where I talked about the day-to-day challenges of living in simplex and how some days are just very, very different from others. At that event, I found out about Rare Disease Week on Capitol Hill that takes place every spring, and how I could go to that event to raise my voice about legislative issues that affect rare disease patients at large. And so I've been attending that event ever since. This past March was my 5th year in a row, Rare Across America. That is another event that's put on by the same organization, which is the EveryLife Foundation for Rare Disease. So I've gone to that event multiple times. I've also done advocacy for the American Academy of Dermatology, the Rally for Medical Research. I've been a patient ambassador for Patients Rising, where I've also shared my EB story. And then with EB Research Partnership, also one of my first forays into advocacy, where I typed in my my story, and now it's on the website where it still lives today. And I've done some similar work with Deborah, just sharing my story broadly with the community. And, again, back to the EveryLife Foundation, as I became more confident about my advocacy journey, I started becoming even more involved in that organization in a leadership role. And I've just found that really fulfilling. And, when I've met other rare disease patients, I've just become so inspired by their stories and how, while our conditions are different, there are a lot of commonalities in terms of our struggles and just the joys that we find from being a part of a supportive community of people who want to make sure that the issues we face and the solutions are well known to our representatives. And until a couple of years ago, there wasn't much information, on EB Simplex or, a lot of treatments for sure. So, in my life, it's been a lot of trial and error. Bandages have improved over the last few years for sure. And then until a few years ago, there weren't really treatments for EB Simplex, but that has really been changing over at least the last 5 years. And so I've actually had the privilege of being in 2 clinical trials. About 5 years ago, I was a clinical trial participant for an oral anti-itch medication. And this was for all EB subtypes, itch being one of the most pressing problems for EB patients with simplex and other subtypes. And so I participated in that trial. And then I also participated in a trial a few years ago for a topical gel that aims to strengthen the skin. And I am so grateful to EBRP for supporting this sort of research. And it's just so nice to see the change from, having no treatments on the horizon to having multiple treatments on the horizon. And then, of course, having 3 FDA-approved treatments for EB as well. So it's been so wonderful to see that positive change in landscape. And, if I were to give any advice to fellow patients, because, just to give back, because I've learned so much from my own fellow patients, one is to, not be afraid to advocate for yourself. Sometimes it's as simple as making sure that your friends and family understand your physical limitations. Teachers, gym teachers as well, that's something that I wish I'd done better, when I was growing up, but of course it's hard when you're in the moment. Talking to physicians about the condition, right? Not just dermatologists. A lot of dermatologists that I saw before I became a patient at the Stanford EB clinic, didn't have much experience with EB. So a lot of times, I was giving them information, but I'm glad that I found the care that I needed. But there have been times where I've had to make sure that they understood that I couldn't have, let's say, a certain procedure, or that I am not able to have adhesive of any kind during, let's say, after a blood draw, for example. So those are things that I've had to make sure that I made known. And then, of course, the 4th bullet is politicians. So that's where I found it really gratifying to tell my story to my senators and representatives and make sure that they support policies that ultimately benefit rare disease patients. And now more than ever, it's a really critical time to do that. And for any other patients who are out there, definitely one thing that I've done that I'm really glad I did was to join EB Facebook groups for just to get additional support. A lot of times we bounce ideas off each other in terms of comfortable shoes, in terms of, let's say, bandaging techniques, or just kind of, voicing, our concerns about what we're going through. And then another point is that really everybody's EB journey is different, regardless of subtype. I was talking to some EB Simplex patients, the other day, and the one thing is that, a lot of times we felt pressured to, think, it's, we're doing fine. It's, things aren't that bad. Others, have it worse. And that is absolutely true. Our condition is not nearly as severe as, let's say, somebody with severe RDEB, which is why I'm so thrilled to see all the treatments that have come out. At the same time, somebody in the group made a really good point that EB Simplex, the skin of somebody who has EB Simplex is also vastly different from someone who doesn't have EB at all. So definitely the skin fragility, it really affects so many things that we do. And so that's something that that I tell, young advocates now, is that, you can have dual feelings about things. You can feel really grateful that, things are not worse, but you can still, wish that they were different. You can still feel really excited about, treatments that are approved, while still wondering, what's ahead on the horizon. And so I think that's an important message. And then, of course, I think, if you're inclined to participate in a clinical trial, it's something I recommend. So definitely stay in touch with EB clinics, with EBRP, with Deborah, and of course, clinicaltrials.gov, so that you're kept up to date on any studies that could be relevant to you. So yeah, with that, thank you again so much, Allison and EBRP, for having me.
Yeah, thank you, Nisha. Your last couple of points especially so beautifully said. So thank you for sharing your story, as always, and for everything you do, not just for the EB community, but the rare disease community at large. We're so grateful for your leadership and so grateful that we could have you with us today. If anyone has any questions for Nisha, like I said, we will address all questions together later on in our Q&A session, but feel free to start to populate them in the chat if there's something on your mind. And with that, I'll pass it on over to Dr. Chan.
Thank you. Next slide, please. So first of all, thank you very much, for having me at this town hall meeting here. And it's my pleasure to present our EBS program and the clinical trial to the community here. Thank you very much here. And this is my disclosure here. And my name is Yiumo Michael Chan, and I'm a senior consultant at TWi Biotechnology. Next slide, please. Today, my presentation will be divided into two parts here. The first, I will describe briefly, the clinical development of the EBS program at TWi Biotechnology here, and focusing on the rationale and the science, of our program here. And then I will provide information regarding our ongoing EBS clinical study called the EBShield here. Next slide, please. For many of, for some of you who know me, actually, I engaged in EBS research at a very early in my career, literally last century here. So, and I had a very fortunate opportunity to discover the genetic basis of different forms of EB, including the EBS, one of the subtype Weber Cockayne, At that time, there was no drug development. The technology for gene therapy or cell therapy or gene editing were not ready, and then we did not have a very good understanding of the disease mechanism here. So for many years, nothing has been done, for the EB. But as Nisha pointed out nicely, next slide, please. Actually, the landscape actually changed, dramatically over the 5-10 years here. So now actually, we have at least 3 FDA-approved treatments for EB, in the last 1 or 2 years here. And this is now a very exciting time, for EB research and the drug development, because we have the critical mass from the patients, from the patient advocacy groups, such as the EB Research Partnership, where we have the industry people, we have a very dedicated researcher like Professor Bauer here. So all together, so we have the critical mass, to move forward the program here. Next slide, please. So now, actually, now let's turn our attention, to the clinical development, or the rationale, of the EBS program here. So our company, TWi Biotechnology, is a clinical stage biopharmaceutical company based in Taipei, the capital city of Taiwan here. So we are focusing on developing treatment for EBS and also other rare skin disease here. So currently, one of our major clinical study is a global phase 2 and 3 EBS study called EBShield. For the drug product here, this is a topical product, and the active ingredient is called diacerein. So you can see the structure at the bottom here, on the left-hand side here. So when the diacerein actually goes into the skin here, it rapidly metabolize into the rime here. So it kind of, the body remove the 2 tails, acetyl tails, on each end here. And the rime is actually a very common natural product, actually is commonly found in many natural products such as rubber here. So next slide, please. And our company actually is using repurposing, or company also say, reposition strategy to develop drug that is in a cost-effective and a time-efficient way to fulfill the unmet medical need in rare skin disease here. And the concept is pretty, straightforward here. It's based on similar mechanism in different diseases here. So all formulation of diacerein has been approved in Europe and Asia for many, many years to treat osteoarthritis here. And this is based on the mechanism that this drug can suppress IL-1 beta, which is a key pro-inflammatory cytokines in many diseases here. So for many years of hard work here, investigators actually, or scientists actually, actually begin to have a better understanding of the EBS and also believe, IL-1 beta plays also pivotal role in the disease here. So those work has been done by, in particularly Dr. Bauer and Dr. Wallis lab in Austria, and also Dr. Joyce Tan's lab, in Stanford University, and plus many other clinicians here. And we basically changed the oral formulation of diacerein to a topical diacerein and the topical form is 1% ointment here, thanks to our founder of the company, Dr. Chen. He is responsible for developing the formulation here. Next slide, please. So how does the diacerein work, or how does the diacerein benefit the EBS patient here? So I would like to emphasize two very important pharmacological activity of this compound here that is highly relevant to the EBS here. So in the industry or in the science, we call this a mechanism of action here. So the first one is an anti-inflammatory action here, just I mentioned, because this drug has been known for many years to suppress the IL-1 beta production here, which we believe is a key pro-inflammatory mediator in the EBS here. As you can see, in many of the EBS patients, in particular, the severe patient, the one on the left-hand side at the bottom here, the blister, quite often you can see a very clear clinical sign of inflammation here. And then the second property is actually the drug can reduce keratin aggregates in the keratinocyte, which is the skin cell in the epidermis here. So if you take a skin biopsy from a patient, in particular a patient from a severe EBS here, quite often you will see some electronic dense material we call toner filament compound here, actually inside the keratinocyte here. So those you can see in the middle figure, on the right-hand side here. So those kind of black structure actually highlighted by a yellow arrow here. Typically, we don't see that in a healthy subject here, the keratinocyte here. We don't see this kind of structure. And also in the patient with more mild EBS, such as the localized EBS here, we also do not see this kind of structure. So we believe those kind of keratin aggregation to some extent correlates with the clinical severity here. So by reducing the keratin aggregation here, perhaps it could also help to reduce the clinical severity of the disease. Next slide, please. Just want to show you, some of the earlier study that shown, IL-1 could be a major factor, in the EBS here. So on the right-hand side here, some of the one investigator from Castella's lab, they took us blister fluid from a EBS patients here, and then analyzed the different cytokines and compare with those of the healthy control here. So immediately they noticed actually IL-1 beta is one of the many cytokines that actually has a much higher level present in the blister fluid in the EBS patient here. Those you can see in the black bar here, and the white bar, the open box here, actually represent the healthy subject here. And then on the left-hand side, actually recently, 2 years ago, Joyce Tan at Stanford, they published a paper. They took the biopsy from the blister in the EBS patient here to try to analyze the gene expression of what different genes, as I expressed in the blister of the EBS patient and compare, with the healthy subject here. They also find that actually IL-1 beta represent one of the very early signaling pathway here, and then through a cascade of signaling, different proteins here is what to actually contribute to the inflammation and blister formation here. So that's the current hypothesis is that in the EBS, the dysregulation of the cytokine signaling creates a viscous cycle of chronic inflammation here, and IL-1 being one of the important cytokines involved. Next slide, please. And I earlier mentioned that actually this drug can also reduce the keratin aggregation here. So Professor Bao and Professor Wally, back in a number of years ago, they published a very key paper here. And they isolate the cells, the keratinocyte, the skin cell from an EBS patient here. And typically you can see under the microscope here, you can see all this punctated staining representing the keratin aggregation here. When the cells are treated with diacerein here, all this keratin aggregate disappear here. We can also quantify actually the number of aggregation or the number of the cell expression, the aggregation here. So those are an independent study that actually we collaborate with Professor Tan actually at a local university, National Cheng-Kung University in Taiwan here. So in a normal cell here, NKC11 cell, there are only very few cells, expressing the keratin aggregation here. But if you isolate the cell from a EB11, which is represent a severe EBS here, you can see a very high number of keratin aggregation. And as soon as the cells are treated with the RIME, the active metabolite of the diacerein here, the number of cells showing the showing the keratin aggregation here are significantly reduced here. And likewise, we can also take a cell, the EB21, which represent which was actually isolated from a mild patient here. We see a fewer number of aggregation here, but yet, when treated with the RIME here, the number of aggregation was also dramatically reduced here. Next slide, please. So our current hypothesis of this drug is this drug can actually reduce the clinical severity of the EBS here. So in the EBS here, we have both local information at the skin, in the blister here, that's mediated by the IL-1 beta, and then the cell inside the keratinocyte here, they have the keratin aggregations caused by keratin gene mutation here. So together, actually, it compromises the skin fragility here, leading to the blister. So when the drug is actually applied to the blister here, it reduces the local inflammation by suppressing the IL-1 beta. It also reduces the keratin aggregation here. So together, although we are not treating the underlying cause of the disease, the patient will still have the genetic defect in the keratin mutation here. We are treating actually, we are making the cell actually, we believe we will make the cell more resistant to mechanical trauma and become stronger here. I think this is the major benefit. And that's another reason why actually we call the study an EBShield study here. So some kind of a protection, for the patient here. And actually, Dr. Bauer actually and Dr. Wally published a very nice paper, actually to describe the relationship between the IL-1 beta and the keratin aggregation through a novel cellular pathway. So we are more than happy, to provide a paper if any of the audience is interested, or you can actually download it from the web site. Next slide, please. And in fact, actually, the drug, the diacerein 1% on has been actually tested in an early clinical study called the delivery study. It's a phase 2 delivery study, phase 2 clinical study here. And at that time, we are collaborating on co-developing with another US company called the Castle Creek Bioscience here. And now Castle Creek Science, focus more on the stem cell therapy, for DEB, JEB here. So basically, the delivery study was an 8 weeks treatment study here, and we actually saw a very promising, results, from this study here that actually, make us, help us to design a better phase 2 and 3 clinical trial, and then also make us believe actually, this drug could have a benefit to the patients here. So as you can see, on the left hand side here. This patient, has a, is clinically defined as a EBS, severe EBS here. When the patient comes to the clinic, the patient has a very severe blister all over the body. And the severity can be measured in 2 different ways here. First, the area of the EBS lesion as expressed by BSA body surface area here. So in this case here, it's about 6% here. And the severity of the blister can also be assessed by a scale called the IGA scale, Investigator Global Assessment Scale. This is a semi-quantitative scale to evaluate the severity of the EBS lesion at a given time based on the size, the number, and other morphological features, such as information here. So I just put together the table. This is a 5-point scale here. So when you have a EBS, when a subject has an IGA score of 0 here, that means basically it's a clear skin with almost no blister or no sign of any EBS here. But like a clinical score of 3 or 4 here represent a moderate to severe EBS phenotypes here with a mix of small, medium, or large blistering with information here. So this patient here also actually show a very dramatic improvement, from the IJ3 at the very beginning, and then after 8 weeks of the drug treatment here, reduced to IJ1 here, as you can see from the picture. Next slide, please. To put actually all this in a perspective of the numbers here. So in the EBS delivery study, we asked the question actually, how many patients achieve an IJ score of 0 and 1, which means a clear skin or minimal blister here during the treatment here. So you can see from the graph on the left-hand side here, the blue line represent the patients receiving the active treatment here, and the gray line represent the patient receiving the placebo treatment here. So you can the treatment effect can be observed as early as 4 weeks here. You can see a difference, a number of the patient receiving the active treatment and showing improvement here. And then at the end of the 8 week, when the treatment stopped here, about 40% of the patient receiving the active treatment show benefit or actually met the success criteria here, whereas only 23% here. And one interesting is this study here, the delivery study enrolled actually all EBS patients regardless of their mutation or the clinical severity here. So in other words, they have patients with both autosomal dominant or autosomal recessive keratin, also some with other non-keratin mutations here. And the clinical severity could be severe, intermediate, and even localized here. So when we look at the cohort, and then we only focus on the severe EBS with autosomal dominant keratin mutations here, we notice actually at the end of the week 8, the end of the treatment here, the number of the patients actually receiving the active treatment actually show actually a much larger difference, effect size here compared to the entire cohort here. So it was about 30% versus the 16% when you look at the entire cohort here. That's why we believe actually the clinical benefit, appear to be more profound in the patient with the severe EBS, with this drug treatment here. So unfortunately, we did not achieve the statistic, of the 0.05, which is actually, always a very important criteria for FDA in the clinical study here. Part of the reason could be due to the limited number of the patient here. So for the entire cohort, we have 50 patients here. For the severe EBS cohort, it was about half of them, 25 patients here. So as a result, our current EBShield study here, we target to enroll at least 80 patients here to make sure we have enough number of the patient to show the statistical difference to demonstrate the effectiveness of the drug. Next slide, please. Here. So based on the clinical experience of this product here, it has an excellent safety profile here. It has a negligible systemic exposure, approximately less than 1% of the oral diacerein here. So that means actually the drug mostly stay in the skin, to treat the blister, and did not actually go inside of, into the blood here. It has minimal local irritation, and the common side effects include application site pruritus and some skin discoloration, but they are all very mild in nature here. And unlike other topical drugs, some topical drugs, they are limited by the application to a certain percentage of the BSA area because of the systemic exposure or other local irritation issue here. So this drug actually can apply up to 50% of the body surface area in adults, or about 80% of the BSA in infants here. So that's about 25 grams per day here. So So which we believe actually is more than enough, to treat the EBS lesion in most of the under most circumstances here. And this product is user-friendly, is once a daily, typically during bedtime, at the evening here. And there's no special procedure or dressing bandage required for the application here. Next slide, please. So yes, let's turn our attention, into the second part, which is the EBS clinical study here. So this current study here, the EBShield study here, is a randomized placebo-controlled design, combined with an open-label extension to demonstrate the efficacy and long-term safety of this product in generalized EBS population here. So that means it's a severe and the intermediate EBS here. So the study will last about 1 year into 2 parts. The first part, the part A here, is very similar to the previous delivery study here. So the patient comes in the clinic up to 10 weeks of screening here. So they will, at the baseline here, half the patient will receive the active treatment and half the patient will receive placebo here. And then at the end of the 8 weeks here, we will have an 8 weeks non-treatment period here to monitor the progression of the or the effect of the disease here. And for the part B here, all patients enrolled in this study here will receive the drug regardless, whether they receive the placebo or the active treatment before here. So that means every patient will have the opportunity to receive the drug product for at least 6 months here. In this study here, all EBS lesions will be treated. So it's not only like unlike many other clinical studies, they treat certain target lesions here, to demonstrate, a proof of concept or to demonstrate the effective drug here. So under our designs here, we will treat the entire EBS lesions here, and patient can continue to use most of the current standard practice of care, but may be asked to avoid certain prohibited medications. So obviously, they are not allowed to take the oral form of diacerein if available at all. So next slide, please. So for the major eligibility criteria here, so as I mentioned here, the patient should have a clinical diagnosis of a severe of the intermediate EBS and confirmed by genetic diagnosis here to be an autosomal dominant keratin mutations in either the keratin 5 gene or the keratin 14 genes here. So about as we know, most of the EBS are caused by mutation in the keratin genes here. And the patient should have certain degree of severity here with an IGA score of at least 3 or about 3% BSA involvement here. The reason is, if the blister are too few and not severe enough, it will be very difficult to assess the improvement in a quantitative way here. And since the product is very safe, we could infants, up to 6 months age here. So anyone from 6 months and above, will be eligible, for excuse me, for this trial here. And for patients with the clinical diagnosis, but without any genetic information here, so genetic tests can be arranged at the site before the enrollment here during the screening period at no cost to the patient here. And one of the reasons actually we will ask is why focusing on the severe EBS, with keratin mutation here. As I mentioned, because of the disease mechanism here, these patients are most likely to associate with the chronic inflammation and keratin aggregation here and show the most obvious benefit under the current trial, under the current study design here. So for patients actually who might have a non-keratin mutation or may have a mild localized EBS here, at this current stage here, actually we might need a different trial design to demonstrate the benefit, of this drug, to this patient here. But we are not forgetting this patient here. So this is just part of a very common drug development process here, one at a time, to do things in a more systematic manner, actually, to show how the drug works and then to get the drug to the market as soon as possible here. So next slide, please. So during the clinical visit here, so patient will come to the site, a designated clinical site, as a scheduled visit here. So the entire study actually will have about 10 visits here, 6 actually at the in the part A, and then 4 visits at the part B here. And then at the study center here, the patient will be monitored for safety and efficacy during the entire study period by trained professionals, mostly the dermatologists here. So for the safety monitoring here is pretty much routine physical exam. And for the blood draw and the urine draw here, it's only limited to about 5, half of the scheduled visit here to minimize the impact, on the patient, because we understand, their skin actually are more fragile. Yeah. And for the efficacy assessment here, these are all non-invasive efficacy assessment here. So the physician will actually evaluate the progress of the disease based on different tools here. So one of the most important tools is the IGA I mentioned in the previous slide here. This is our primary endpoint to measure the success of the trial here. We will also, the physician will also determine the body surface area of the EBS lesion to see if they improve or if, or not. Yeah, and then there's another scale that actually I did not describe here, it's called the EBA-Darcy scale here. Yeah. And the patient also have the opportunity to document how they feel and how they function by completing some quality of life questionnaire, pain scale, and the pruritus scale here to describe, how this affect, their daily life here. So other things, including taking photos of the lesions and then keep a diary to record the drug usage and the use of other medications, management of the skin lesion during the study here. So next slide, please. And how do we define the success, outcome of the patient? What do the patient will expect in this clinical study here? So when the patient come in, actually the clinics here, they will have blister here. And for this example here, so they maybe have a IGA score of 3 and 4 here. So if the drug works, after 8 weeks of treatment here, the patient who are receiving the active treatment here should show some improvement, to IGA of 0 and 1 here. This is how we define the success of the the study here. And in fact, the number of the patient receiving the topical diacerein should and showing the improvement should be much higher than the number of the patient who receiving the placebo here. Then after the 8 weeks treatment here, during the open label, the 6-month open label period here, because all patients would use the product. So we expect if the drug works here, they will show continuous and sustained improvement during the 6 weeks here. So next slide, please. So since EBS is a rare disease, recruitment is always a challenge here. So that's why we actually need to have many different clinical sites in many different countries here to recruit enough patients for this study here. So currently, actually, we have more than 30 clinical sites in many countries, literally in 4 continents here. So this is truly a global study here. And in the future, we might add more sites and countries here, depending on the current recruitment progress here. Next slide, please. So we target to enroll 80 to 100 eligible generalized EBS patients, in this clinical study here. And US is a major center or major, site in our region, for this clinical study here. So we have 9 currently sites, across 4 different time zones, in the US here. And we are making so far very good progress, in this study here. So far we have randomized, about 30 patients here. But your continuous support and participation, in particular the US, is very critical, for this study here. We greatly appreciate your support. Next slide. Yeah. So finally, actually, to conclude, my presentation here, EBS has a high unmet medical need here with no approved treatment at this time here. So as a sponsor here, we have tried everything we can actually to move forward the program, to help the patients here. First, we make sure we have a very good understanding, of how the drug works, what I mean by the mechanism here, and then the product is safe to the patient here. So, and then actually we design a clinical trial in a way that actually, to optimize the chance of the success, and in this case, we target our EBS population to the severe and the intermediate EBS with autosomal dominant keratin mutations here. So for the drug approval process here, we work closely with FDA here, and we have already obtained, several designation and voucher here, and those are actually, with the purpose to help to accelerate the registration approval process here. So we are very excited and optimistic, about the recent progress, in the community, in the EBS research here, and we feel we are, very honored to be part of the story, in this exciting time. And next slide, please. And obviously, we have to thank you, all the KOLs, our clinical advisors, for this program here, because without their efforts here, I don't think, we will have this program, or at least, we will not moving, this fast here. So we have a wonderful team of the KOL, to guide our program here. In particular, I would like to thank, Professor Bauer here. He actually, he lays out all the foundation, for the diacerein research, to treat EBS here. And then Joyce Tan is our first author and one of the leading PIs in the deliveries trial here. So we published the deliveries study results 2 years ago here, and then we are more than happy, to provide the paper, or you can actually, download, from the web here. And then Dr. Spellman was the CMO of the Castle Creek during the delivery study here. And Dr. Chui Jiao Kai and Dr. Malario, in the UK here, they are KOL, in rare skin disease, in particular, EB research area here. And next slide. I would like to thank, everyone. There are so many people to thank, actually, but actually, in particular, I would like to thank, the EBS patient and their families, and the PIs for the participation and support in the previous and the current EBS study here. And I would like to thank Allison and Michael, at the EB Research Partnership, for actually having us, to present our program and study here. Just the final slide here. For more information, about our clinical study, the EBShield here, please visit. There are many ways, actually, different ways, to receive the information. You can go to clinicaltrial.gov here. You can either type in the exact number, or you can type in, EBS, diacerein. You can also visit our company's website, or email directly to us. You can also contact, Allison, for more information here. So, my last, actually, before participation, in a clinical study here, please, discuss with a physician, to determine if this is the right study for you, and understand the risk and benefit of participation in a clinical study. And as a patient, you have absolute right to withdraw from the study anytime for any reason. It's completely voluntary here. So thank you very much, for your attention.
Thank you very much, Dr. Chan, for, taking the time to talk us through the trial today, the science behind it. It was incredibly informative and educational. I know I learned a lot. I think everyone on the call did too. So thank you again, and to TWi for all the amazing work being done. Margot and our team has been popping those links in the chat if they're a little bit easier to click on or copy and paste. So you can go to the chat if you'd like to use any of these links. And finally, I'd like to pass it on over to our final speaker of the evening, Dr. Bauer.
Yes, thank you very much also to Dr. Chan for this very comprehensive overview about the story behind diacerein. Just to reiterate a few things here, and to give you a feeling why we think this is an effective drug for patients suffering from EB Simplex. For my side, and it has been mentioned already, I'm now working about 2/3 of my career that I spent in investigating EB. I was working on diacerein, so I'm working now since 15 years on this story. Initially, we did some basic research, and we found out this might work in a topical preparation. And in addition to the study that Dr. Chan has mentioned, we did 2 early on initiated studies in Austria and in France and Czechia at this time, which were also very positive. So that basically laid the ground for the DELIVER study then. We think it's a good drug and it's a safe drug. I can say that from the clinical experience. There is no major side effects. We have not seen allergies to diacerein when it has been used in the daily life of many patients, because in Austria patients get it for free basically, or have gotten it for free. Now it's blocked to facilitate the study again. So we have really good experience about this topical preparation. In terms of some practical things, Dr. Chan mentioned already, it's applied just once daily, and usually we recommend to do it in the evening, after a bath or after cleaning wounds and making a fresh coverage of the wounds. You apply the cream to the wounds and a little bit outside of the wounds, but that's just per protocol. We can study the protocol for the study. And then you cover the wounds as you do it every day. There's no specific new coverage that you use. You should not use very highly absorptive coverage. So the cream should not be absorbed into the wound coverage. It should stay on the wound. So that's something you have to consider. One thing also, which is probably important, but that's certainly discussed with your doctor at your clinical site, is that it has a slight yellow hue. So there is a possibility that you get staining on your beddings, so that has to be considered. And certainly in the initial phase, when there is a placebo, you will also have a yellow placebo, so you will not be able to discriminate it from the color. That's an important point also in the study then. Yes, so it's once daily. Should be good. There should be a good feeling with this cream. You should not apply it to the head and genital area. So anything else can be used as the treatment area, but you should spare the heads basically. So that's basically my real world experience, my experiences we have with this cream. And as I said, we're using that since basically 13 years, 12 years now, and it's really doing well. Not every single EBS patients does respond. Certainly, we have to be very careful to include the right patients this time into the study. Dr. Chan alluded to that. We should focus on the dominant forms of the keratin mutations. But we do see other patients in the simplex area with other genes affected, where it's basically also effective, but that's not within the study. So we have to be careful about the inclusion criteria here. And we certainly also have indications that it might be effective in recessive dystrophic EB, but that's another story that we are covering then in later phases of the work that we are intending to do. Okay, so that's basically my short statement to this study. Thank you very much, and I'm open for questions.
Thank you so much, Dr. Bauer, for that perspective, for joining us today. I know it's quite late where you are, and just thank you for everything that you and the EB House make possible. We're so grateful. With that, we're going to move on to the Q&A portion of our discussion today. Just as a reminder, we're going to try to get through as many questions as possible, prioritizing what was pre-submitted, although I do see some questions in the chat. Don't worry if we don't get to you. We will personally follow up with you and get your question answered via email. So our first question here that we have is for Nisha, Dr. Bauer, and Dr. Chan. I think you've all alluded to this in some different ways, but someone was asking, how have you seen the EB landscape change since you've been involved with the community?
Sure. Well, I'm happy to start. I would say that when I was growing up, it didn't even occur to me that there could be a treatment for EB. You know, it just felt like science fiction to me. You know, it's something I couldn't even conceptualize. And you know, to think that now there are multiple studies. First of all, a few FDA approved treatments, which just blows my mind. And for EB Simplex, there are several studies currently underway. Is just, it's really heartening. So that's just, that kind of progress is great to see. And then also even, aside from the clinical studies, I feel like there's just more knowledge in terms of dressing wounds. The bandages are much more sophisticated than, including, let's say, Mepilex, than what I grew up with. So it's great to see that, that sort of improvement as well.
It's just we have the same, feeling that about 30 years ago we did not even know what the genes are that are affected in patients that are suffering from blistering all over the body and on the mucosal surfaces. And we were very happy 10 years later then to get diagnosis by doing sequencing in the respective genes. But then it took another 10 years to make the jump from diagnosis to getting first hints for treatment. And certainly one of the first approaches was stem cell gene therapy that we did with Professor Michele De Luca from Italy, where we did transplantations in the 10 years ago, which went very well for junctional EB, which did not work so well for dystrophic EB. But no, we are happy that 10 years later, Abiona did it and got the approval for this stem cell transplantations for recessive dystrophic EB. So it's really exciting that many of the old procedures and ideas that we had are coming to fruition at this point. So it's really exciting, as Nisha said, and so we are looking forward to more that's on the plate.
Yeah,
I see a question here. May I say about the question, because that was
in the
directed
chat
to
about
me.
the EP house?
Yes,
Yeah,
yes.
I think that would be great context.
Just to stick on that, EB House Austria is the national reference center for EB patients in Austria and neighboring parts of surrounding countries like south of Germany, Italy, and Hungary, and like Serbia and Croatia. We are attached to the Department of Dermatology at the University Hospital there, and the kind of prominent feature and the good feature we think that's prevalent here in the EB house is that it's separated from the clinic. Basically, it's attached, but it's separated, and our doctors and our nurses and our researchers are just working on EB. So it's not a small part, or it's not something we do in the afternoon for our patients. But we are really doing nothing else there. We have 1 or 2 patients per day, and we're happy that we can afford that. We have good funding from the patient organization in Austria. So we are really dedicated to the issue, and that's a good thing that we are able to do this. We have attached research. We have what we call an academy, where we educate medical personnel how to deal with epidermolysis bullosa. So people are coming from all over the world, basically from Taiwan, from Australia, from the US, from Brazil, to get educated. And we also have a very active clinical trial department where we are focusing on the newest trials that are around globally. And we're also, designing new trials, new investigator-initiated trials. So anyone who's interested how that works is welcome to come to Salzburg, and we're going to show you how this all works. Thank you.
Thank you so much, Dr. Bauer. Michael and I were actually lucky enough this past October to get to do exactly that and go and see how it works. And it's an amazing center. So thank you for talking us through that. And thank you to Jeff for pointing that out. Our next question is for Dr. Chan. This person said, I saw the EBShield study already has a lot of locations, but if my medical center wanted to become a location, is there a process for that?
Sure, more than happy to answer this. So actually, we have a very thorough, and diligence, process to evaluate medical site and to open a clinical site, for clinical study here. So in addition, actually, we also have a patient referral program, to help patients, to direct to other medical sites. And also we have some travel and reimbursement, to make it easier, for the patients here. So basically, if anyone wants to actually find out more about the location, where actually they can join the clinical study here, they can either contact, Allison, or they can actually, contact us, at the email address here, and then we'll try to actually provide the information, or at least to direct, to the source, who can provide the information here. So basically, we'll try everything we can, to accommodate the patients, to find the site. Thank you.
That's amazing. Thank you so much, Dr. Chan. Maybe one last one for Dr. Bauer. I know there's some stuff going on in the chat. We're, like I said, taking a note of that. I'm happy to connect anybody with any of our speakers if they're comfortable, or just find an answer to your question and get back to you after the town hall. So maybe just our last live one. Dr. Bauer, someone is asking if gene editing or gene therapy is also being considered for EB Simplex.
It's certainly considered because, the newer gene editing technologies are able to change the mutated base in the keratin 5 or 14 gene. But the price is higher than with recessive dystrophic EB and even junctional EB, because there is not too much scarring going on in EB simplex, and therefore all the procedures that are attached for example, to cell and gene therapy with skin transplantations are very harmful for the patients. And so from a clinical side, I would say this is not the way to go, and we probably have, really soon a good local therapy, but that is not so harmful, meaning that we probably have diacerein gel to be used then. So I have to say, yes, we are working on that, and other people are working on that. But I think from the clinician standpoint, I would prefer for my patients to have some topical cream ointment for the patients.
That's a really helpful perspective. Thank you so much. And just a big thank you again to all of our speakers for taking the time today to go through their presentations and answer these questions. We'll make sure that if your question was not answered, that we send you an answer shortly after this. So before we wrap up, how can everyone on this call support the life-saving efforts that we heard about today and really accelerate EBRP's mission to cure EB and pave the way for everyone battling a rare disease? We have a couple of ways that you can get involved. Firstly, as you may have heard, Matter of Time, a unique rock documentary capturing the electrifying energy of Eddie Vedder's October 2023 solo concerts in Seattle that were part of our Venture into Cures event, will be premiering at the Tribeca Festival in New York City this June. We're so excited for the world to see this film that blends together powerful music with the moving real-life stories of patients, families, researchers, and thought leaders that reveals how determination and innovation are paving the way towards a cure. And we're launching a raffle that gives you the chance to be among the first to see the film by joining us at the premiere. I don't know if anybody tried, but tickets sold out in about 30 seconds. So this is a great way to be able to see the film along with all the families and researchers that were in the film, and to join us in June. So we're going to drop the link in the chat here. This is a raffle run by our friends at Fandium. So you enter by making a donation, which will benefit EBRP and help advance our mission. The raffle closes on June 1st, so be sure to enter before then if you're interested, and good luck. Also for our folks in the San Diego area or any Major League Baseball fan, we've got some exciting news and a good event for you as well. Games between the San Diego Padres and the Seattle Mariners have been informally termed the Vetter Cup for a couple of years now, as both teams vie to be Eddie Vedder's favorite hometown team. And now the rivalry is real. The Vetter Cup is an actual trophy to be won. The first series of this Vetter Cup will take place at Petco Park, with May 17th being the official themed game. So if you come, your ticket includes a themed item and a donation to EBRP. So if you're in the area or willing to travel and a big baseball fan, this is a great way to show your support and really to have some fun too. So we'll be putting the link to that in the chat as well. And lastly, our next town hall, as we've been doing, will be at the end of next month. That'll be at the end of May. We're still finalizing our date and speakers, so be sure to keep an eye out for that announcement. And to please subscribe to our newsletter if you haven't already. That'll just allow you to stay in the loop on not only our town halls, but on all of our exciting updates. You can subscribe to that newsletter at ebresearch.org. And that's all we have for you guys today. So thank you so much for taking the time to join us. And again, to our incredible speakers, but really to our audience as well, thank you for being here to learn from them and to ask these questions. We hope to see you next time. And I hope that everybody has a great night. Or if you're a part of our dedicated Taiwan crew joining us at 5, 6 a.m., I hope you have a great rest of your day. Thank you everyone so much.